people in the waiting room. Yeah. Okay, are your slides there? Do you want to put them up?
You're supposed to join in, but I'm not joined yet. So
Can you let her in? Can you let Tatiana in? Go to the waiting room, go to view. Go up there. All the way to the right. No, don't touch any of that. Go to the right. That says 10 people.
no,
going. Go this side. Yeah. Go to view.
it says view.
Okay, and then I think at this point you can admit everybody and then you can get started. Just say admit all. Then you will also be in.
It says the meeting host will let you in soon. I have to change some settings with my
Go ahead and admit all because we'll just tell you that way you can start playing with your.
yeah.
Or spell.
All right.
L
No.
L T at different time. And
Well, so Ollie said there's a simpler way to do the audio.
OLE.
Because you don't want me to use any of my audio. See, I don't want to do this. I don't want to.
I think you just stand here. And
She wanted me to use the audio system.
yeah, I think
like-
it'll show up in this. So
Oh, okay.
just let's ask people, can you hear us? Can anybody hear us? Just give us a chat and say if you can hear us. Yeah. so they can hear us.
Okay.
So I think you just want to come here and share your screen. Turn off all the. Okay, good. Yeah, I think so. We'll just move the. And then I think now you get to do it. Then we'll see what.
Somebody in the,
Ollie, how do you do the display? How do you change displays?
change displays as in, so I see it. I see the,
You see you see it
shared
as the
screen.
audience? Okay,
yes.
great. Okay, great.
Yes. And if somebody down there would be able to scoot the lectern forward just a couple inches, that will help a lot with our being able to hear her clearly.
Can you hear me?
Yeah, try one more time.
Can you hear me? Volume louder?
That's good.
Oh,
I think.
good. That's loud. Okay.
Can people hear? Tatiana, just say, yes, you can hear her. It
you
needs to
can't
be a bit louder.
See.
There's a camera there, so they're watching you through that. So maybe we bring you closer to one of the mics. Is that?
All right, stop. Stop.
Yeah, yes.
All right, how's that? Good.
I think that's great.
Oh, good. Okay.
Okay, let's get started. Everybody, thank you for joining us online as well as we're in person here. We're going to keep track of everybody joining online. I'm really pleased to have Professor Tatiana Schnur come give us our first 2023 seminar for the Center for Brain Recovery. Tatiana is an associate professor in the departments of neurosurgery and neuroscience at Baylor College of Medicine. I've known Tatiana for a long time now. We all study, we're all interested in the brain after it has a stroke, and she's been doing some amazing work looking at recovery right after a stroke. So without further ado, thank you, Tatiana, for sharing your work with us today. And the way we do this, this is the first time you're joining us, Tatiana will talk for about 45 minutes. If you have a question, you can post it in the chat window. And then when we're done, we'll sort of alternate between people in the audience as well as people online, and we'll try to moderate that. So thank you, Tatiana.
Okay, I just want to check. It looks like people might be seeing the back of my head online. So I do that, then you see me from the computer.
Yep.
Yeah, so I think we'll do it that way. Although I'm sure that the back of my head is not very tight. So first of all, Swati and everyone here, thank you for inviting me to come share my work. This work should be quite familiar to you in terms of its importance and why we're doing it. Oh, that.
You're going to keep track of.
Sure.
Yeah.
I'll have to go.
Okay,
Keep going.
yeah, just keep going and we'll keep admitting people.
Okay. So. You're sure? We're good?
Yeah.
Okay. So before I begin, I bet it's. Not advancing. On the screen and then.
You can't move your screen?
Nope. Oh, there we go. I think it's. There's like a super delay on the, like when it advances.
Okay, we'll just keep trying and then let's see.
Okay, so
That's not happened before.
let me, yeah, okay, we're good. So I think we're good. All right, so before I begin, I want to do, so this is good because this is an introductory slide. So if we have further technical issues, we can certainly address it right here. I want to tell people about how I got into this field. I don't think people talk about this often when they begin their talks. Like apparently, I think this is a little bit sad in terms of diversity, but like many academics, I am in academia because one of my parents was an academic. And so technical issues. Specifically, my mother got her, and there's a delay again. My mom got her PhD from Georgetown in linguistics in 1977. And her PhD was looking at aphasia and semantic impairments. And so that just means that by high school, I was doing science fair projects, looking in linguistics and language, actually language acquisition. And then in my this talk is going to get more complicated, though.
Trying to move.
Yeah.
Yeah, and it might be on your laptop, but not on the share
No,
screen.
nothing. There we go. There's one. So we got one. The title of my mom's dissertation was a psycholinguistic study of the semantics of aphasic language. But then, I think it was my senior year of high school. I think it was pointed out to me in the Washington Post. So I grew up in the Northern Virginia area. I saw this article. Neurology Human Backup Systems for Language. And this was a paper published in Nature in 1991, and I thought it was amazing. It was 2 single case studies, and it showed a double dissociation in writing and speaking for grammatical class, like nouns and verbs. And I just thought this concept, that you could have localized damage that would specifically target a function was amazing. This work was done by R.G. Hillis and Alfonso Karamazza, was published. And that's what got me into cognitive science, was reading about this paper. I subsequently went and got my PhD from Alfonso Karamazza years later. And just like people in this room, I have collaborated with R.G. Hillis. So it's sort of full circle, but I wanted to give you a sense of like how I came into what I study. So this won't be foreign to you guys, but almost 17 million people worldwide suffer a first-time stroke each year. And it's really great. We have improved interventions like TPA, and mortality is declining. But what that means is that people, the brain damage from stroke is affecting the ability to produce speech. And I think the latest survey I saw is around 30% of people who have a stroke, a left hemisphere stroke, are going to have language impairments. And a pressing issue, I think, that we're all really interested in trying to solve is why some individuals with acute language production deficits recover really well, and others continue to have moderate or severe deficits in connected speech. And so I just wanted to give you an example of some of the differences in speech outcome we hear over time in what we've tested in our lab. So for example, 3 days post-stroke, this is someone we tested in the Texas Medical Center. She has a basal ganglia infarct, and this is what she sounded like when she was describing this, doing picture description, and this is the picnic picture from the Western Aphasia Battery.
Just speaker on your laptop?
No, because of the double.
Yeah, just go.
Would you like to?
I forgot to ask you when you're sure it sounds. I can switch the sound over real quick.
Yeah, that would be.
So you, I'll turn the sound off here first, and then I'll let you know when you can unmute yourself and unspeaker your speaker.
So just a second, folks, we're gonna switch the speakers. Yeah. In the.
Thank you for sticking with us here. I'm
Shoo. Okay.
Can you hear Tatiana?
Can you hear me? Any chat box? All right, good. All right, so now maybe we'll try it. This isn't actually overall that important in terms of my talk, but I wanted to give examples of things that we've heard when we, there we go. Basal ganglia
You
stroke.
can go ahead and you can hold that if you want. Yeah.
Okay, well, let's see. A man and a woman are having a picnic. The man is reading a book. He has his shoes off. She's pouring a drink, it looks like from a thermos, and the thermos is the hamper is open, but I don't know why. There is a transistor radio, and I guess that's all I can say about that one.
Okay, so I'm gonna, so people can still hear me, right? We're good. Okay, so I just want to point out, like, she sounds really fluent, right? But she's got some disfluencies, and it looks like also she's either doing circumlocutions or doing low frequency substitution. So that's someone 3 days post-stroke. Then we have someone who's a month post-stroke with a much larger left temporoparietal infarct.
Can you tell me?
The whole Combear?
Mm-hmm.
The whole Combear is a, I would say, a family situation ground that have chair made on a keef with shell made to carried things around a blanket.
So that person sounds very different, right? Maybe a classic Wernicke's. And then we have someone who we tested 5 years after their stroke.
Remember this?
Yeah.
I want you to try to tell me as much as you can about what's happening in that picture. And try to talk in sentences if you can.
Okay. Trees. Kites. Boat. Book. Sandwich. Listen. Coffee.
So that's the end. That ends the audio portion of this talk. Before I continue, do we want to change anything on this?
You know?
Yeah. So I can stop sharing and try it again, maybe. Do I mute myself at this point, or do we?
I think this,
sharing the changing.
we changed the sound settings. I don't see a reason to.
No, but project it, share your screen.
Okay.
It was just, we saw the.
Yeah. All right. So now we're going. Okay, good. So, great. So what I want to highlight here is we're interested. Oh my gosh. Zoom quitting. Okay, good. All right. Going. Oh, okay, good. You're right. Great. So I think the question that those examples highlight is like, who recovers, who doesn't? How can we predict patterns of language production deficits after you have a stroke? And something that I'm particularly interested in that came from my graduate training going forward is how we produce speech and how does it get damaged? That is the sort of theoretical organization of language. So many of us, and for many years, we talk about single word production. So we're able to produce 2 to 3 words per second out of a spoken vocabulary of 40,000 words, and we only make a mistake one every 1,000 words. How do we access this information? How is it organized? What is the information that's used to produce speech? So these are questions I've been very interested in, but, you know, we don't speak in single words, we communicate with each other. And so there's been a movement to trying to get to more this question of connected speech. And so how do we produce multi-word utterances? And what are the brain regions that are required for the successful execution and recovery of connected speech? So that's what I've been focusing on in my work more recently, is trying to get at a better sense of how do you proceed with connected speech and how does it fall apart after you have had a stroke, and then what are the patterns of recovery? So left hemisphere damage, we know, often impairs language production. This is a meta-analysis by Kathy Price from 2010, and it shows, you know, a left lateralized network, but a lot of those data came from comprehension or single word production. So the processes for producing and organizing the content into structure within that network are not well identified for connected speech. And so there are reasons for this. I mean, there are reasons why we focus on single word production, confrontation naming, is it stems from the difficulty in both eliciting and measuring spontaneous speech. It's just a lot harder when, especially, you know, ideally we're going to get into conversations and dialogue, because this is how people use language. So the first thing I'm going to talk about in my talk is work that came out of the late teens, where almost all brain lesion behavior mapping analyses of connected speech in speakers with stroke has been limited to analyzing the content and the speed with which it was produced. So i.e. the words elicited from like picture descriptions or direct questions. So in the past 3 years, but a few more papers on this topic. But for example, people will describe a scene like the picnic picture or the cookie theft from the Boston Diagnostic Aphasia Examination. And you can use measures like the number of words. These are easier to analyze. Fluency, words per minute, or a qualitative score. Very interesting in general, but not related to syntactic structure, or combinations of words like the lexical variety or the informativeness of speech. And so the first bit of research I wanted to talk to you about was addressing this question of, are unique brain regions required to produce the content and structure of connected speech? The work I'm going to talk about today is done in collaboration with a postdoctoral fellow who was in my lab. He's now at the University of Edinburgh, Junwan Ding. So I just want to point out that he is on both of these papers. And then moving from identifying the brain regions that are required for connected speech, what does recovery of connected speech look like in the year after stroke? And can we identify any acute predictors? And so this first step in this direction was looking at acute damage to white matter in the acute stage of stroke, which in this case was in 3 days after stroke, which some people refer to as hyperacute stage. So, why are we interested in, content's very interested in connected speech. Well, what about the structure? So trying to understand the regions that are supporting structure and whether structure and content really dissociate speaks to a central debate about the specificity of brain regions required for syntactic processing and for the stages of connected speech production where content is combined into structure. So that was really the impetus here, was trying to get more at not just the words they're saying, but how well are people combining them into bigger structures and their morphosyntax. So to do this, we enrolled 90 participants for the first time acute left hemisphere stroke. We enrolled from 3 comprehensive stroke centers in the Texas Medical Center. So I'm Baylor College of Medicine, and this is a 10-minute walk to 3 stroke centers, and now a 4th one has just opened up. So we're able to go at bedside, consent people, and test them. So we enrolled 90 participants, and we tested them within 4 days of their stroke. And we had to exclude 25 because they couldn't complete the task. And you might ask, well, why should we look at acute stroke at all? A lot of people we look at are people who are suffering from chronic aphasia, chronic stroke. Well, one, you can examine brain behavior reorganization before brain behavior before reorganization of function. So people start, I think, reorganizing the minute after they have a stroke, but we're at a very early stage to try to map brain regions with behavior. Also, I really want to highlight here, because this is different from other studies, we have a continuous enrollment of acute stroke participants. So they are not enrolled on the basis of an aphasia diagnosis. It's simply whether they've suffered a first-time left hemisphere stroke and they don't have other reasons that might explain their language impairment. And what this does is it increases variability of behavior. So sometimes we get people with aphasia, sometimes we don't. Also variability in terms of lesion size and lesion location. By examining people with chronic stroke and their communication, they sometimes have severe impairments because they have damage to a lot of regions at the same time. So if you want to dissociate function of different regions, if both regions are damaged, you can't know what each one is doing independently. So we're mapping brain damage to performance, connected speech. So we quantify brain damage using a gold standard, which is having people delineate lesions on a scan. We collect our scans. This is part of what's acquired clinically. So clinically, we get diffusion weighted imaging, CT perfusion if people are contraindicated for the neuroimaging, for the MR. And then you use diffusion weighted imaging because this sequence is the most sensitive to the infarct. And so you can see that hypointensity here. Is that there? Yeah, okay. And so what that's reflecting is what strokes do is they affect the water flow. So what you get is a sodium potassium channel breaks down. You have water outside the cells that flows into the cells. And so this is indicative in the early stage of tissue that is dysfunctional. If they're contraindicated, we delineate the lesion on CT perfusion and we co-register it to a T1. So we tested our patients within 24 hours of their clinical neuroimaging. So there should be a good link between their performance and what looks like to be dysfunctional tissue as a result of the stroke. And then we create lesion volume. So someone, in this case, my collaborator is Chris Hamilton, is delineating the lesion on each slice, and you create a 3D volume. So each person has a 3D map of where we think their damage is. And then we use lesion behavior mapping. And in this particular study, we use multivariate LBM, but for, I'm not sure anyone in this room, but maybe there are people watching this talk, they aren't as familiar with this. So lesion behavior mapping learns a pattern between damage and behavior. So you have each, so there, like each rectangle is a person's lesion volume. And for each person, you have some behavior you've collected. And then you want to know whether if a voxel, which is a 3D, I think it's about like 50,000 neurons, supposedly this 1 millimeter cube of brain, if that cube of brain gets a 1, it's damaged. It has a 0, it's not damaged. And then you can ask, does damage in these particular groups of voxels predict one set of performance versus not worse performance versus better performance? And then you get a series of beta weights, so voxels that are tagged with numbers suggesting that there's a better link between damage and performance or a worse link statistically. Okay, so that's how we got the brain part. Then how do we assess connected speech? So we used a storytelling task. So it doesn't matter which story you use. In this one, we used a common story like Cinderella. And we asked people, they viewed all the pictures in the book and the text was included in the book. And then after they refreshed their memory about the fairy tale, they closed the book and we asked them to tell us the story as if we had never heard it before. And measuring storytelling is on the step. It's providing better ecological validity with abilities to produce everyday speech. I don't think that this is completely analogous to walking around and talking to your friends or family members, but it's a step closer to trying to get at spontaneous connected speech. So we had, let's see. We're gonna try, this is a short sample of someone describing the fairy tale at bedside.
See, we saw the story of Cinderella happening in the hallway. She was instructed to repair or replace the
So that just gives you an example of someone trying, not that you guys could hear it in the room, but online, someone telling this story in the acute stage of stroke. We were, for this analysis, we had 65 narratives that we transcribed, and we used quantitative production analysis, which is a lexical syntactic approach to dividing up connected speech, assessing different capacities in connected speech. And this approach comes from Safran, Elizabeth Rochon, Myrna Schwartz and colleagues. Jean Gordon has several publications about this as well, using this approach. So in the quantitative production analysis, you get a bunch of different variables. And some of these, and this is either measuring lexical or syntactic aspects. And these variables can be correlated with each other. So to reduce the dimensionality, we categorize a connected speech using principal components analysis. And this particular analysis yielded 4 components. And so now I'm going to describe what these components were because I refer to them for the rest of the talk. These 4 components explain 71% of the variance from the narrative. So the first one, sorry, and this is in 65 people. The first one we called structural complexity. And this one had these 5 different variables that were part of this component. And we, looking at these variables, I mean, that's the thing with PCA, you have these variables, you bring them together, but that, factor 1, what do you call it? So it looks like mean sentence length, mean utterance length, elaboration, the degree to which people could elaborate phrases, embed clauses, This all seems to point to an ability to produce words in increasingly complex combinations, which requires individuals to arrange words based on appropriate thematic and or syntactic roles among other constraints. And to just give you an example of like, well, what is one of these variables? Phrase elaboration. It's the length of the subject noun phrase and the length of the verb phrase. So this is something someone told us: the wicked stepsisters went to the fancy ball. The length of the phrase is how many elements are in the noun phrase, and in the verb phrase, how many elements are in the verb phrase. So you'll have a better elaboration index the more that you can put into phrases, and you'll have a worse one if you have fewer elements. Okay. The second component was what we called lexical selection. So this was the proportion, it goes in two directions, the proportion of nouns you produced in comparison to closed class words, pronouns, and verbs. But in the other direction, you could be producing a lot of pronouns, verbs, and closed class words in comparison to nouns. And so for those of you who don't know what closed class words are, you go and mark up for every utterance how many closed class elements there are compared to open class, like verbs and nouns like went and ball. So here we call this component lexical selection. So the ability to produce words with grammatical functions like pronouns, verbs, and closed class words in comparison to nouns and vice versa. So that's the second component. The third component we called syntactic accuracy. And so here the variables that loaded on this component were proportion well-formed sentences, auxiliary index, proportion words in sentences. So a sentence is classified as having a noun and verb. So you can have an utterance that only has a noun or only has a verb. That's not a sentence. Sentences can be ungrammatical, but you just have to have a noun and verb, or you can have this proportion. These all seem to load together. And so we called this the ability to generate, we think of it as morphosyntactically accurate speech. How well are they doing that? So an example of a well-formed sentence is the prince found the slipper, but a not well-formed utterance would be they could not put it. And then the last component was loaded on a single variable, and that was words per minute. And so this seemed like the speed at which connected speech is produced. So I'm going to be talking about these four aspects of connected speech: structural complexity, lexical selection, syntactic accuracy, and fluency. And so we conducted 4 separate lesion brain mappings to see where brain damage best predicted poor performance across these 4 components. So the first, I should add, of those 65 people, 52 had scans that we could do the tracing on. So sometimes in those CT perfusion scans, the quality just isn't good enough for us to be able to delineate brain damage. So in 52 people, we did 4 of these lesion brain mappings. So what do we find? For structural complexity, producing less complex structure, like fewer words and utterances and phrases, fewer embedded clauses, this was associated with damage to posterior temporal cortex and angular gyrus in these people with very little reorganization after their stroke. In contrast, syntactic accuracy deficits, like omitting required noun determiners, verb arguments, or morphemes, were associated primarily with damage to the left inferior frontal gyrus. So this result in of itself is a little bit different than what people find in comprehension evidence, for example. And I'll speak a little bit to that shortly. What about lexical selection? Producing fewer nouns in comparison to those other groups of words was associated with damage to the middle temporal gyrus, posterior superior temporal sulcus, and the angular gyrus. That's what's showing up in yellow here. But in contrast, producing fewer words with grammatical functions like determiners, prepositions, and pronouns was associated with damage to regions near but not overlapping with those associated with syntactic accuracy deficits. We have this, so we have this clear dissociation, at least in this particular patient group, lastly, production fluency, damage to multiple regions like pre-central gyrus, post-central gyrus, inferior temporal sulcus, inferior parietal lobe, some subcortical structures. This was associated with impaired production fluency, and this was simply just the words produced per minute. So this one question, like if we're trying to understand someone's ability to produce a content when they're telling a story versus organizing that content into structure, it seems that these things dissociate. There are different brain regions that are critical in some level for these two aspects, these abilities to produce connected speech. I was interested in, do these results map onto theories of connected speech production? And I'm not going to talk at length about this here, but I did want to point out, so this is a model that's based off of speech errors from like Fromkin at Yale in the 70s and onward. But Bach and Liefeld published a paper in 1994, and they outlined some different stages that they thought happened during connected speech. So you have this message you want to convey, and let's go out to lunch. But this message has to be aligned with the concepts that you know, and then you have to retrieve the words in the particular language you're speaking, and then organize those words into structure. So this first stage they called functional processing. So here words are lexically selected and potentially with the grammatical class that they're associated with. And for the heads of phrases, you might get the number of arguments. And so to convey event structure, they argued that functional encoding also, here words are associated with each other via thematic roles. So what someone does to someone else or something else. And potentially this, although this is not clear, you might assign syntactic roles here. So those can divide. Whether you're the subject or object of a sentence is different from the role that you might play in that event. And so based off of this principal components analysis and the quantitative production analysis, I interpreted that the middle temporal gyrus and the posterior superior temporal sulcus were regions required, right, for the successful lexical selection and this generation of lexically driven argument structure. Because remember, it's a combination of words into larger and larger phrases or the embedding of clauses. And so this seems to map onto functional encoding. In contrast, positional encoding is argued to be where you position utterance elements in order. And so the syntactic realization of these lexical semantic structures and their linear ordering, you have to mark those with bound and freestanding closed class morphemes. And so this might be based in the LIFG and the insula. So regions necessary for producing words with grammatical functions and accurate morphosyntax overlap in these two areas. So we have this rostral caudal dissociation between content and then organizing it into structure. And it is consistent with syntax evidence in other clinical populations. So direct cortical stimulation coming out of Eddie Chang's lab in primary progressive aphasia. And just one example of this is work by Steve Wilson. There have been some more recent papers, which I'll talk about at the end, that also support this interpretation, but it's not consistent with sentence comprehension evidence, which it's mixed, but seems to involve both frontal and posterior regions associated with syntactic processing. I think it's, and you have work both in from this group as well as across the water in Evelina Fedorenko's lab. Very interested in this topic about, do you really need separate neural structures for syntaxes that exist in comparison to just combining things. So we then moved on because we collect longitudinal data. We test these people at bedside, and then we bring them back at multiple time points in the year following their stroke. And so we are interested in understanding the patterns of connected speech recovery based off of those 4 elements I just described, and whether there are biomarkers that might predict different patterns of that recovery. And the first one we looked at was white matter damage acutely. Are certain tracks when they're damaged predictive of certain connected speech function? So there's been work elsewhere that suggests that the degree to which regions are connected contributes to connected speech performance, and at least with general cognitive abilities, connectivity may better account for impairments in comparison to lesion topography. And so we looked at the tracks that connected regions that have been involved in language. So what we found, what I just talked about, was of course frontal IFG damage and areas near the temporal parietal junction. And so we looked at the tracks that connected these regions. And we also looked at tracks connected with the anterior temporal lobe, but we don't see any ATL damage in our sample, and that's because that tends to happen with larger strokes. And at bedside, the people who are able to produce a whole narrative generally don't have very large strokes. But nonetheless, there are some connections that are frayed with the ATL, and so we looked at the uncinate fasciculus, which connects the ATL with IFG, and if you're a longitudinal fasciculus. So we looked at the tracks that connected these regions, and I'll talk about predictions shortly. And we looked at longitudinal recovery of connected speech. And we assessed damage acutely and then tried to predict patterns of recovery. So what did our longitudinal patient enrollment look like? So people we tested acutely within 2 weeks who are able to come back at least one time point, there are 41 of these people. And then, so these groups all were tested. So we tested them subacutely. Of those 34 people, all 34 were tested acutely. They just may not have been tested at all other time points. And that's why they're different. These are overlapping groups of subjects. It's about 80% overlap of each group. We also did a subsequent analysis which supports the results I'm going to talk about in the subset that were seen at all 4 time points, and that was 20 people. So we assessed connected speech using the storytelling that I talked about, and these 4 components of connected speech. And then we, to talk about what the pattern of recovery looks like. When I say someone is impaired at baseline, we decided, we elected to define baseline as they're doing worse than 1 standard deviation lower than controls. So that's worse acutely. And so the most frequent impairments in acute connected speech, and those people who are able to produce measurable connected speech at bedside acutely and who are able to return at follow-up, remember that's our sample here, was reduced words per minute. So fluency, 25% of these people had problems with fluency. And then it was about 10% of people had reduced ability to produce nouns in comparison to other types of words, decreased syntactic accuracy, and structural complexity. So that's just what, in our sample bedside, that's how these people looked in their connected speech. Who improved? So in what aspects of connected speech? So the proportion of patients who improved from that baseline impairment, for fluency, 50% recovered to the normal range. But what's quite surprising is these are people who had, I would argue, mild impairments. Half of them are not improving to control levels a year out of stroke. And I want to point out here that the light blue is performance subacutely, so within 1 to 2 months of stroke. The middle one is 6 months, and the last one's 12 months. Now for producing nouns in comparison to other words, in contrast, 80% eventually recovered within the normal range for lexical selection. So these 2 were the components that were sort of worst off in terms of recovery. In other aspects, everyone got to within normal range. So in syntactic, having morphosyntactic elements, well-formed sentences, everyone got to 100%, as well as for structure. And this is different. We were not screening people for aphasia, and I'm guessing that we don't have the more severe people here because they were actually unable to complete the task when we tested them at bedside. Okay, so those are the patterns of recovery. Is there a marker acutely that would predict these patterns, or at least correlates with it? So we estimated track damage, but we didn't have high resolution scans or sequences. We had to use what we had acutely. And so what that means is we had our acute lesion mass, which I described, that's just simply where their lesion was in their brain. And then we had a track mask, which was the probability of where their tract was based off of work from Roykova and colleagues in 2015. So we overlap these to understand how much tract damage there was acutely. So an estimate of damage to a tract based off of the acute lesion mass. And then what was the analysis path? We looked at the degree of change. So we subtracted acute and subacute performance. So how much better did they get from your acute baseline? How much better did they get at 6 months compared to acute baseline? That's early chronic. And late chronic is 12 months later. How much better did they get 12 months compared to acute? But as people may be aware of, baseline performance correlates very highly with degree of change. And that's, partly because if you start off really low, you have a lot of room to improve. But if you start off higher, there's less change. So we regressed out baseline performance, and we also regressed out total lesion volume. So you could imagine, well, if you have simply more damage, then you may recover less. So we factored out these two. I do want to add, in our sample, and this is in contrast to some other studies, total lesion volume did not correlate with almost any metric of change. It did correlate with performance acutely. And I think this is because they're just smaller strokes. And then what we did is we grouped people, either they had damage to the tract, which was estimated at having an overlap of greater than 100 millimeters cubed, or they didn't have damage to the tract. And then we asked if these two groups differed in the degree of recovery for a particular connected speech variable. And we did a non-parametric t-test to determine this. What were our predictions? Our predictions were based off of what we saw in the first paper. So for fluency and syntactic accuracy, this was based off of damage to the frontal temporal cortex. So we expected that anterior connectivity between the IFG in terms of the frontal ascending tract and supplemental motor areas should be involved, and also with frontal areas and posterior areas like the supramarginal gyrus is connected. The IFG connects with the anterior segment of the arcuate fasciculus, and middle temporal regions, which is the long segment of arcuate fasciculus. And I will add, we had predictions that connections posteriorly should be related to word retrieval and structural complexity. So here, the posterior segment arcuate fasciculus and the inferior longitudinal fasciculus, ILF, should be related to these two components, to the degree of recovery. So if you have damage to these tracts, you're going to recover worse, because these functions are associated with temporal parietal junction or posterior superior middle temporal gyri damage. And here I'll say that one of our predictions bore out and the other didn't, just as a preview of what I'm about to show you. Also, we controlled for multiple comparisons. So we were concerned that although we had these predictions, I wasn't strongly wedded to them in a sense. So we controlled for multiple comparisons. So the results I'm talking about surpass a threshold. So what we found was that the frontal ascending tract seemed to be very important for recovery. So here what you see in red are people with damaged tracts. And you can see that the change in their fluency scores at the acute versus subacute stage. People in blue had preserved tracts. And what I'm going to talk about here is a preserved frontal ascending tract. And we found that acute frontal ascending tract damage predicted worse fluency recovery at the subacute stage. And then for fluency, it didn't predict subsequent patterns. So your degree of recovery at 6 months or 12 months did not seem to be subserved by this FAT damage. So this was consistent with what we expected. What we didn't expect was that FAT damage also predicted worse recovery of structural complexity. So if you had damage to this anterior connection, it predicted recovery of structural complexity, which we had shown was subserved by regions posteriorly. So here you can see that early chronic, the change in performance from acute to 6 months later was predicted by how much, whether you had damage to the fat. And this stayed through the late chronic in 12 months as well. And we think here, and this is a post hoc explanation, that there is reorganization. And so maybe there are frontal regions that are taking over some of that function about combining words into phrases that you don't see early on. We replicated these results in people who were seen across all 4 time points. So both in the sample where some people were in some groups and not others, and also when they were seen across all 4 time points. So, take home message for this part. And the second part is frontal ascending tract seems to be critically necessary for better recovery of connected speech. And this was, as far as I'm aware, the first longitudinal study of lexical syntactic recovery of connected speech. Caveat in that there are 41 people. It'd be much nicer if we had 200 people. So we're working on that. But it was the largest study that I know of. So in summary, my lab conducts longitudinal studies of language recovery from acute through chronic stroke. And I talked about identifying the brain behavior organization of connected speech. So posterior regions seem to be critical to select words and combine them into structure. And frontal regions seem to be critical for syntactic processing. And we identified those who would benefit from early intervention. It's a first step in this. Reduced frontal connectivity predicts poor connected speech outcomes. Before I end, I just, we're doing other work in the lab, and I just wanted to briefly point to it. We've been working on new methods. So we do big data connected speech assessments, and it's a real pain to actually analyze this stuff. And so we're trying to think of ways to make this easier. So one was a collaboration with Vita Fromm and Brian McWhinney at CMU using their chat clan. And this was creating an automated quantitative production analysis approach of connected speech. That has really, and I should say it's like semi-automated, but it has really eased things. Also, for those of you who study single word production and confrontation, like naming errors, it was getting really tedious, not arguing, but discussing repeatedly whether someone's speech error was semantically related, associated, unrelated. And then if you look at verb naming, it's even like a bigger problem. So in a recent paper with a former postdoc in my lab, Cha-Ming Lee, who's now a professor at the University of Radford, we used word2vec, which is like distributed semantic model to assess naming errors, and then collected independent tests of lexical semantic access in comprehension, and found there was a high correlation between the assessment of semantic deficits, like this, the degree of semantic similarity between someone's word error and the target, and the degree to which they were failing on these independent tests. So I think that means head out, go forth and use word2vec if you want to stop arguing about semantic errors and picture naming. And lastly, we also are interested in relating brain damage to behavior. If you have 2 patterns of brain damage that predict performance, how do you know that they're the predictive validity, right? Like, are they independently predictive? And so does one set, if you have damage here, definitely mean you're going to have this performance, and damage in another pattern mean you will have it, you don't. So we use predictive validity to compare associations between brain damage and behavior. This is work with John Magnotti, who's at the University of Pennsylvania. And we developed a GUI. It's available on my website. If you have your lesion maps, and if you have 2 behavioral sets of scores across a bunch of patients, you can then test the prediction that the maps are different. I have a whole set of work with my collaborator, Randy Martin at Rice University, looking at cognitive capacities that underlie connected speech, like working memory and lexical selection are really maybe a function of executive control. And then lastly, we got a new grant this past summer looking at function in acute right hemisphere stroke patients. And so we're looking at whether theory of mind impairments are related to communication impairments. And as I wrap up, we couldn't do any of this without our participants and their caregivers. So thanks to them. A thanks to the neurologists at the hospitals and everybody in my lab, and of course NIH funding, which made this possible. Thanks.
Ollie has a plan.
Okay, thank you so much. We are gonna take 5 minutes and then we're gonna take questions. If you want to take questions.
Yeah, that'd be great.
We're gonna, the physical people, we're gonna move upstairs. Digital people, just hang on for 5 and we'll see you out there.
Yeah.
Who's moderating the questions? Hi.
Sophie gave me the order to moderate questions. If anybody else would like to do. I'm gonna say, you have a question for Tatiana, and either just shout it out or put it in the chat and we could read it out for you, or use the hand raise function. No, there's going to be plenty of questions.
Tatiana, I just want to say thank you so much for your talk. I do want to chat with you, and I think I'm going to get a chance to chat with you later, but I actually have another meeting that I have to head to right now. So I'm so sorry. I'm going to hop off, but I think I'm going to get a chance to chat with you later. So
Excellent.
I look forward to it. But thanks for a great talk.
Thanks.
All right. Bye.
Not sure if there are any questions with our pause.
Yes, go ahead, G.
I'll repeat it.
Yeah, if you get closer to the speaker.
Yeah, sure. So I found your talk is very interesting, and I have a couple of questions, but only two now.
Okay.
So the first question is, it was very interesting that the features and complexities catching up the recovery very quickly and very higher functions measures. So I wonder whether it's because of the syntactic processing or but it's more like automatic. It's taps onto more automatic cognitive things like based on the working. You might be familiar with the automatic processing is more related to the syntactic versus or working memory.
So hold on, although I'm not sure. I think there are two people holding on here who might be listening to this, so that just so I understand, the first question was that people who had deficits acutely in the ability to combine words into phrases and also to produce syntactic, morphosyntactic structure seem to get to ceiling or whatever, but within the range of controls by 12 months, and the other like fluency didn't. And maybe one reason for this is that there might be another capacity that's subserving it, right, that helps it get better. It's entirely possible. I mean, I think we don't know. We also had a smaller sample of people, and there was a little more variability in the syntactic performance. Some work with Randy Martin points to the fact that working memory we didn't make predictions about syntax, but we made predictions about that ability to combine words into phrases under the, conception that lexical semantic information that you might hold in working memory, you need that working memory in order to generate longer phrases. And the smaller your working memory is, or if it's impaired, you're not going to be able to generate longer phrases. And we did show that in the acute stroke sample. We did not find any associations acutely with working memory and syntactic performance, but it's something we're interested in because you would make the prediction that the people who improved, maybe it was their working memory, their working memory came online to support the syntactic performance. So there should be later a correlation between working memory and syntactic performance if you don't see it acutely, right? Some reorganization of function.
There was a very interesting, just what you said reminded of what working memory actually does for making the semantic and lexical units more richer to make, for them to produce longer sentences versus the people who produce the same length of the utterances, but fewer closed word classes who have depending on the working memory. So if you're going to compare those two, then I think those are two different, components of the system.
We found no correlation between what we called semantic working memory and the production of closed class words. Mm-hmm. I don't know. I think it was around 50 people acutely. But I think reorganization is probably quite a lot that happens.
Other question is about cross-linguistic. So I found your work on the principal component intriguing. And I wonder whether there is any evidence that the other language actually have produced any evidence, those kind of
components. Great question. So the second question is whether when you divide up the lexical syntactic variables in quantitative production analysis, you do it in English, you get, and this work, by the way, was replicated, like Jean Gordon showed this as well. It's sort of, and the original paper by Elizabeth Rochon, much smaller samples in chronic stroke, show that similar clustering of variables into these components. And the question is, what about other languages that maybe don't have linear ordering, or, however, what does it look like? So I'm not aware of, there might be results out there that if tried to apply QPA to other languages, but
studying, try to translate.
Yeah.
I didn't have enough data to run a principal component analysis, but I used this component, and the data were high. So complexity, fluency. But yeah,
the reason I was wondering about that is that, depending on the, the components that we can find, kind of fact, like So I think that's basically what I was interested in, like because linguistically, the brain has been ways, the way that they learn from different language structures. And yeah.
That's really fascinating, right?
Yeah, that's gonna be very.
But I think the way you divided the component based on the principal component analysis makes sense, right? Even without running the principal component analysis, I would divide the component, right?
Yeah, some shift,
Yeah.
but yeah, it also makes me feel better.
Yeah, maybe I wouldn't necessarily have two structural components because you had syntactic complexity and you had also,
Structural complexity.
yeah, maybe I would.
I think
just
some
say-
people would have argued but they didn't hang together, right?
Yeah,
That's
exactly,
right.
exactly.
In
Yeah,
this particular, in this sample.
that was the
Yeah.
only like surprising. But the rest.
So it's 2:10. Does that mean that I formally close the talk? Okay. Thank you, everybody. The last Aaron and Andrea.